which also used to be called Chlamydophila pneumoniae in the past, chlamydia comes from cloak, and pneumoniae means, well, pneumonia. So chlamydia pneumoniae are bacteria that hide inside host cells, particularly cells of the respiratory tract, and they cause pneumonia in humans. Now, chlamydia pneumoniae is classically considered gram-negative because it can't retain the crystal violet dye used during gram staining. Additionally, it has an outer lipopolysaccharide membrane, which all gram-negative bacteria also have. However, other gram-negative bacteria also have a thin peptidoglycan layer under the lipopolysaccharide membrane, whereas chlamydia pneumoniae doesn't. Without it, chlamydia can't retain the pink safranin dye used during gram staining, so they're not technically gram-negative either, which is why they're more correctly called atypical bacteria. To visualize them, a Giemsa stain is required, which makes them look blue or reddish-blue. Now, chlamydia pneumoniae is non-motile and round-shaped, and is also an obligate aerobe, meaning that they depend on oxygen for survival. Additionally, chlamydia pneumoniae are obligate intracellular pathogens, which means they are unable to generate ATP, so they rely on other cells for energy. When outside of a host cell, chlamydia pneumoniae are metabolically inactive, so this means that they can't grow on artificial mediums, but rather require a host cell for culture. Okay, now when C. pneumoniae enters a host cell, it undergoes a life cycle that alternates between two distinct forms. The first is the small, spore-looking form called the elementary body, and it's the infective form of this bacteria. After the elementary body enters the host cell, it gets enclosed in a vacuole called an inclusion, where it transforms into a metabolically active, star-looking form called the reticulate body. The reticulate body can use the host cell resources to divide, and it does that by binary fission, which means every reticulate body splits into two identical copies of reticular bodies. On a side note, if this sounds similar to mitosis, well, it is. But the term binary fission is used to describe division of prokaryotic cells which don't have a nucleus, and therefore some steps in replication are different from mitosis. Now eventually, binary fission results in a huge number of reticulate bodies, which then start transitioning back to elementary bodies. So the cell becomes too small, bursting open and letting out a lot of elementary bodies in the surrounding fluids, where they attach to other cells to repeat the cycle over and over. Once in the lungs, the elementary body is taken up by epithelial lung cells inside an endosome through phagocytosis. Inside the endosome, the elementary body transforms into a reticulate body and begins to replicate. The infection causes both sustained airway hyperresponsiveness and inflammation, which increases secretions in the airways. Chlamydia pneumoniae typically causes a mild form of pneumonia, also called walking pneumonia, but it's also able to get into the bloodstream and can even cause atherosclerosis. What is more, elementary bodies may also be released into the world as respiratory droplets when a person coughs or sneezes. If another person breathes in these droplets, they get chlamydia pneumoniae as well. And the risk of getting the infection increases with age, so it's one of the leading causes of community-acquired pneumonia in the elderly. Now, individuals infected by chlamydia pneumoniae are often asymptomatic, or may have nonspecific symptoms like fatigue, sore throat, mild fever, and dry hacking cough, all of which aren't typical of bacterial pneumonia, hence the name atypical pneumonia. Besides, the person may not feel very sick, as opposed to a person suffering from other bacterial pneumonia, where they'll be surely bedridden and suffering from more severe symptoms, like dyspnea or shortness of breath, fever, chest pain, and a productive cough. This is why sometimes a case of atypical pneumonia is also referred to as walking pneumonia. However, these symptoms may be more severe in the elderly. Now, a microimmunofluorescence test, or polymerase chain reaction, or PCR for short, is the preferred method for diagnosis of chlamydia pneumoniae. The microimmunofluorescence test detects serum antibodies against chlamydia pneumoniae antigens, while in PCR, a sample like a pharyngeal swab is taken to find chlamydia pneumoniae DNA. Chlamydia pneumoniae pneumonia can be treated with antibiotics like azithromycin. Alternatives include tetracyclines, like doxycycline, but they're contraindicated in children and in pregnant women. Alright, as a quick recap, Chlamydia pneumoniae is a non-motile, round-shaped bacteria and an obligate intracellular pathogen. Chlamydia pneumoniae undergoes two changes during its life cycle. There is the infective form or elementary body which infects, and the reticulate body which replicates. It's observed with Gamsa staining. The symptoms of Chlamydia pneumoniae pneumonia are usually sore throat, mild fever, and dry hacking cough. The diagnostic gold standard is a microimmunofluorescence test, and the first-line treatment is azithromycin.